And then just to let you know, we still have 2 openings before the Maze meeting. Several folks did sign up last week, but we still have openings on February 7th and February 21st. So if you got that wonderful news from Ruben that your talk has been accepted, or if you got poster material that you'd like to practice before the group too, those are wonderful opportunities to get ready for the Maze meeting. So I'm going to keep reaching out to folks over the next day or two to see if we can fill next week's slot, which is February 7th. I'll message in the channel if we haven't filled that spot and if we're not going to be having Z Evolution next week. But please log in. This is a great forum to present your research, and please go ahead and sign up for a talk. Okay, great. So today we are pleased to have Yuebin Wang from the National Key Laboratory of Crop Genetics and Improvement at Huazhong Agricultural University. And she'll be presenting her research on a spatial transcriptome map of the developing maize year. So I'll go ahead and turn it over to Yuebin. Thanks a lot.
Thank you. Hello, everyone. It's very glad to be here to introduce you one of our project about a spatial transcriptome map of developing maize ear. And my name is Wang Yuebin, and I come from China. I come from Huazhong Agricultural University. Firstly, I will introduce you some background because as we all know, people keeps increasing, and 800 million people are still hungry every year. So I think the biggest problem we all face is to making sure everyone has enough food in the future. Maize is one of the most important crops. We all know it's grown a lot and also gives a lot. Therefore, improved maize yield is really essential for the food security. But we also know maize yield is a complex quantitative trait. So people breaks into different factors to understand it better. Its yield comes from the ear number per acre, kernel number per ear, and kernel weight. So very obviously, we can find the genes that control the maize ear development is very important for increasing its yield. So in the recent and maize ear, we can see it's arise from the inflorescence meristem and its development organs, including the spiculate pair meristem and the spiculate meristem and floret meristem. And currently, many mutants linked to the meristem transformation and development have been identified. So the study of this mutant also have greatly advanced our understanding of the genetics and the molecular mechanism behind the ear development. And 3 years ago, and a research team that led by the David Jackson, they used the single cell sequence technology, analyzes the cell type within the maize ear, and successfully identified 12 cell types. And we can see this milestone marks the beginning of the single cell age in the study of the maize development. And in addition to the single cell RNA-seq, in recent years, the spatial transcriptome technology has greatly enhanced the cell type annotation and the single cell analysis. And very notably, impressive progress has been achieved in some species like human and mice. So we wonder whether this technology could be also used to achieve a better study in the maize ear. So currently, I can see most spatial transcriptome technology is based on the frozen sections. Therefore, the first step we can say is to obtain the completely frozen section. Hence, in here, I will show you some field cases field cases from us, and so we can see the hardest part of this frozen section is the cell in the section must not be damaged, and the cell shape must be normal, and I can see it's very difficult. It's a very difficult part. Secondly, we also experienced many I can see many challenges trying to figure out the timing of mRNA released and the condition for tissue removed. The most difficult part is that the mRNA appear diffusion or the mRNA is not released sufficiently. In the most ideal section, we finally obtained the nuclear accident staining result showed that after the tissue was removed, the mRNA barely diffused and was completely fixed in field. It's very difficult for us to obtain this result. And at the same time, the mRNA is also fully released. So based on this result, the experimental result, we are using the BGI's stereo-seq technology to construct the spatial transcriptome of the maize ear. We harvest about the 6 millimeter stage ear of the B17.3. I can tell you why we chose this stage, because in this stage ear, it's containing the most meristem type. And finally, we can obtain the high quality spatial transcriptome map from the 4 independent sections. You can see the result is very good. After comparing the 4 sections, we found that 10 cell types could be repeatedly detected, which including the 4 cell types that never been reported before. For example, here I can show you we discovered the 3 cell types in the inflorescence meristem. The 3 cell types that have never been reported before. This including the inflorescence meristem tip. It's IMT here, which contain in the top about 4 or 5 layers of the IM, and inflorescence meristem pharophyry, which along the outer pharophyry of the IM, and the inflorescence meristem, which in the central region of the IM. To further validate the sensitive and the occurrence of the stereo-seq, we randomly selected some marked genes and some well-checked genes that identified by the stereo-seq, and conducted the mRNA in situ hybridization experiment. In here, I think please pay special attention to the 2 genes on the rightmost side. You can take a closer look. It's the best result we have got, because it's evident that the stereo-seq currently capture the gene expression pattern, and we can finally validate the spatial expression pattern in over 70% of genes. And we next asked if the stereo-seq dataset could help us in understanding how stem cell determination, because it's we care about this question. And therefore, based on the spatial information, we can now extract the spots from the indeterminate and the determinate meristem for reclustering, and obtain the 3 subcluster. These 3 subcluster were clearly distributed in the different domains, which including the inflorescence meristem tip, determinate meristem pharophyry, and the determinate meristem tip. You can see this in the screen. We also detected a series differentially expressed genes among these 3 subcluster, which including the 2 very special genes. It's 2 maize box genes, ZM maize 8 and ZM maize 14. These two genes were specially these two genes was a specific expression in the determinate meristem tip. You can see they also expressed in here. And according to the previously study, we know that the maize family genes contribute to the cell fate determination. We therefore generated the knockout mutants of the two maize genes. In the double mutant, we observed the florid meristem were converted to the indeterminate year branch, which means they are unable to differentiated into the next level organ. This indicates that the two maize box genes are the key regulator that determine floret identity from stem cell. So with the special, so now I think we can get the precisely position of the cell types. So now we can construct a developmental trajectory of the year based on its real structure. And actually, we do observe the two major trajectories. One is a cell in the meristem outer edge. This cell from meristem outer edge transition into the meristem base. And another is the cell distributed in the upper vasculature differentiated into its base. So this result demonstrates that at least two developmental trajectory in the maize ear. We can see here, one is a vertical differentiation and another is a lateral differentiation. In addition to mining the spatial transcriptome data, in this study, we also introduced the real single cell data. So the next part, we focus on how to integrate the single cell data and the spatial transcriptome data. In here, we're using the deconvolution algorithm, and we can directly annotate the half of the cell type from the single cell RNA-seq data. And to further verify the result of the integrated result we have got, we also identified the cluster-specific mark genes in the single cell RNA-seq and verified their expression pattern in the stereo-seq data. In here, we can see at least 5 genes supported for the prediction of the spatial information for each single cell cluster, and which also demonstrate the reliable of our integration result. And by using the annotated single cell RNA-seq data, we next identified the 3 cell type specific genes, specific gene modules, and a series of hub genes in the co-expression network. Furthermore, we can use the spatial information to examine the expression pattern of the hub genes and the co-expression network surrounding the hub genes. Thereby, we can obtain their potential functional locations. You can see we obtain the 3 very specific expressed gene modules in here. And more importantly, compared to the randomly picked genes, we discovered that the hub genes showed a higher heritability for year trait that related to the yield. This suggesting that those hub genes very likely contribute to the maize production. In here, I want to say, because in the 3 gene modules in here, module 7 and module 3, actually it's a well-known gene modules in the maize ear development research. And in this module 5, we can see the NOC this is the transcription factor, and we can see this TF is the hub genes in these gene modules, and we also generated the CRISPR mutations to verify this gene functions, but we still not have the transgene result, but we are focused on this gene in the future. So, in summary, I think in this study for the first time, we applied the spatial transcriptome technology, which is the stereo-seq on the maize ear, and resulting detailed clarification of the cell type. And we also have the precisely position of the cell types. And through extracting and distinguish the subtype of the meristem cells, we identified the two maize box genes that control the stem cell determination. And also by integrating the single cell RSeq data, we identified the 3 cell type specific gene modules and a series of spatially specific hub genes in the co-expression network, which I think is providing the novel genetic resource and the method for maize breeding. So this is the content this is a presentation I today. And in conclusion, I would like to express my thanks to the Professor Yuebin Wang and the Professor David Jackson for their invaluable guidance. And I'm also very deeply thanks for the colleagues from BGI. And I also am truly appreciative for the opportunity granted by this conference to share my work with all of you. Thank you.
Thanks so much, Yuebin, for a great talk. I've been super excited about single cell technologies, and it's fun to see how you guys are using those data. I posted a question in the chat while other folks are thinking of their questions. And I was just curious if you knew how reliable some of the marker genes that you identified are across developmental stages. Can you always rely on those marker genes at various different time points?
Mm-hmm. Okay, I know. Oh, sorry. I will show you I will continue showing my screen. Can you see my screen?
Yes.
Okay. Actually, it's not because the stereo-seq has its limited. I think all of you can understand that because it's a very new technology. But so in here we randomly picked some genes. It's not a well-characterized gene. So and we also picked the well-characterized genes. It's have the in suit hybridization experiment, and we verify the two expression pattern with the stereo-seq and their hybridization in suit hybridization result. And we we do observe that some genes that not very consistent. They are not very matched, but in the last we found over about 70% genes, 70% genes, they all match the expression pattern and the stereo-seq result. So I think it's a reliable result.
Great, thank you. And Josh, looks like you have some questions as well. Feel free to unmute and ask those.
Great, thanks. Really nice talk, and I saw your preprint out on this, and it's really nice work. One question or a couple of questions I have are mostly technical questions. And so the data that you showed here, that's based on cryo sections. So have you tried this with using fixed tissue? Because I know that for other spatial transcriptomics approaches in plants, fixed tissues and paraffin sections is what works as well. I don't know if you tried that with the ear or not.
Oh, yeah. I understand you. You mean the fixed method we use is it right?
Yeah, well, I guess I'm confused. Are the data that you showed, is that on sections from cryo, so frozen sections, or are those fixed and then embedded in wax and then sectioned?
Sorry. Oh, sorry, I don't understand. Can you repeat again?
Yeah, I'm
You-
sorry. So the sections that you have, the sections of the tissue, are those cryo sections, or are those microtome sections from fixed tissue that has been embedded in wax, like paraffin?
Oh, you mean the wax? Oh, sorry, we don't try the wax.
Okay, that's just
It's
curious.
all the fresh. I mean, the wall section we obtain in the last is the fresh is fresh tissue, and they through the freezing.
Okay.
You know what I mean?
I do,
The
yeah.
freezing.
That was just a sort of technical question. I was, yeah. And so the other thing I was just wondering related to that then, I guess, is do you freeze the tissue, immediately after dissecting it, or is there something that you put it in as far as like a cryopreservant or something like that?
No. We actually we in the last we think the freezing time is the most important. So we only try the freezing method, not
Great.
another method. I mean, to try to fix the tissue.
Great, thanks. I might circle back with another question, but I'll let someone else jump in with a question.
Okay, thanks, Josh. Jeff, it looks like you've got a question as well.
Yeah, great talk. That's super cool data. And this is probably something that you folks are thinking about, but I wondered if some of the MADS genes you've identified, or the hub genes, if you've looked to see if those are things that show either differences in expression or evidence of selection between maize and teosinte.
Okay, I understand. Actually, we are trying to construct the thousand year spatial transcript data, and we got some results, and I didn't show you here. But I think there is absolutely some genes exist in the maize year, and I mean, you see the selection between the maize and the thousand. But because we just got the thousandth year spatial transcript data just maybe a few weeks ago, so we didn't analyze it. But I think it's a very good idea. Maybe we can I mean, combine this technology with the evolution.
Yeah, I think that'd be cool.
Yeah, and I'll just chime in there too. I mean, the hub genes would be interesting. It might also be interesting to look at the genes where you see kind of rewiring of those networks between maize and teosinte, the ones that either come into the networks or fall out when you compare maize versus teosinte, and then look for signatures of selection in those genes.
Yes. And actually, I sorry, because you spoke of the maize and the thousand, actually, I have one question for Jeffrey. Because we can see the maize year development, it's very different from the thousand. And I wonder which part you think the biggest, the most different part do you think the maize and the thousand year have? It's a core. I mean, is the curve or the year number per row something?
I'm not sure how to answer that. I mean, there are a number of differences, and as someone who's pretty far from being a developmental biologist, I'm not sure that I'm the right person to answer that. Ruben or Josh or somebody, you have thoughts?
Sorry, I actually was multitasking on something else. I missed the beginning of the question.
Which parts of the ear show the greatest difference between maize and teosinte? Is it development in terms of row number, or the cob, or?
Actually, I think it may be the cob, but I don't know if I'm right.
Yeah, I don't know. There's several structures there that sort of come together for the whole. It's hard to say in developmental time and space. But yeah.
Okay, I understand. Because now we actually we need a break. I think we need a point to figure out what's the difference between the maize and the teosinte ear. I think the most different part of the two species may be the corp, but the ear number is also very important, right?
Yeah, and that kind of brings up a question for me too. I mean, are there very many studies out there where they've done like careful comparisons of teosinte and maize developing ears and identified homologous tissues across those and done these in situs?
Yeah. And another very important question is we didn't know the development stage of the teosinte ear.
Yeah. Josh, do you know of examples like that where they've done side-by-side comparisons and looked at homologous tissues and done hybridizations?
Not that comes to my mind right now. No.
Okay.
Mm.
So you might have
Yeah,
a nice frontier
mm.
Yuebin here to take a look
Yeah.
at.
Can I ask one more quick clarifying question that's more technical?
Go for it.
Okay.
So the individual sections that you showed, are those individual ears as far as like what we would think of as a biological rep, or are those different sections like serial sections within the same ear?
Sorry, what?
Sorry, you showed two different sections of your tissues. Are those different ears, or are those different serial sections within the same ear?
Oh, it's different ear.
Okay, great. So then I was wondering, across your biological replicates, how often did you find some of the same profiles being,
Actually, in our study, this section is the same year, but the two sections, it's a two sections from the same year, and this is the different year. So actually, we obtained the 4 sections from the 3 different year.
Okay.
Uh-huh.
Yeah. Again, all these things are sort of new methods, and I just was curious what you're sort of landing on this finding out, because it's expensive, right? So you don't have a lot of choice to do a lot of sections
Yes.
and things like that. So
yes, you are
yeah.
right. It's very expensive experiment.
Yeah, so some of these early studies are really sort of quite informative, not only in the biology but just also the approach. So yeah, thank you.
Thank you.
And Elliot, looks like you have a question as well. Feel free to unmute if you'd like to ask that.
Yeah, thanks for this. This is a really cool project. This was an awesome presentation. I think because making these sections seems really difficult and sequencing these seems really difficult. One thing you could do to get a sense maybe of the diversity across maize lines is check out some of the genetic differences in your marker genes and like the hub genes and related genes to see how much variation there might be in like publicly available genomes. I'm sure some of these are like better annotated than others and like better defined than others, but even just looking for like, are there a lot of stop codons or like a lot of coding sequence variations? Like you could imagine if you had found an accession that only had one of those 2 MADS-box genes functional out of the 2 that it takes to like knock out to lose the differentiation, you might like not as a hub gene or as like an important gene. I think you could like pick up maybe some things that you would be potentially missing in these networks, but also kind of see which of these are like actually like essential or like a big part of being like maize versus teosinte if you end up looking at those differences. Because it's like really possible that there could be like variation in like variation. There's like some SNP that's varying in both teosinte and maize, and you get like one version in maize and a different version in teosinte, but those might actually be like driving the difference in the morphology. I just think that it seems a lot cheaper than trying to section and sequence more lines. So it could be like a quick way to go about that.
Thank you for the suggestion. I think it's a very good question, and also it's very important for us. But as I said, the spatial transcriptome technology is a very expensive experiment, and we also apply this technology on B73. But yes, I think maybe we applied in another lines, no matter the maize box or the hop gene. I think we can find more variation, but not now, maybe.
Yeah, I think you probably don't have to do the sequencing to look at the variation though. Like you don't have to do the spatial transcriptomic sequencing because the public genomes are available. So if you just go check out like, are the genes like broken? Like are the coding sequences broken in other lines? Then you can like avoid actually having to do the spatial transcriptomics to find that.
Okay, yeah. Okay, thank you.
So Ellie, what you're saying is basically to help quality control some of these marker genes or something like that, you could look at patterns of variation rather than having to do the experiments.
Yeah, exactly. If that's
Yeah.
something you're interested in. I mean, this is so cool already. Like it's not like you need to do that, but it would
Yeah.
be a cheaper way of answering those like
Yeah.
teosinte versus these questions.
I think that's a great
Yeah.
suggestion, Ellie.
Okay, yeah, because I want to say here, because this data set we also published as the resource, as the genetic resource. I think it's maybe very useful for the development for the research for the studying the maize year development, and we also find some genes that we now have the transgenetics experiment. I think our aim is to find the genes, not the data, but our aim is to find the genes that control the maize year development. So yeah, I understand you, but that we are trying to work on this.
Yeah, and thinking about this, and Ellie, you got me thinking it would be interesting, once this isn't quite as expensive, to do kind of the core dispensable type analyses that Candy Hirsch's group has done a lot of on a single cell type, basis, and look across the diversity of maize and identify cell type specific core genes and dispensable genes. And yeah, that'd be a lot of fun. Ruben, looks like you've got a question.
Yeah, I was wondering if, keeping in the in the vein of looking at the genetic diversity, if somebody has looked into variation in the patterns of expression in the either in the developmental trajectories or in the tissue patterns of expression of some of these genes.
In the tissue, what? Sorry.
Yeah, no. So some of these of the genes that you identify here, I was wondering if somebody has looked into whether the same gene is more expressed in a particular tissue type in one genotype versus another, and whether that variation in the pattern of expression can lead to differences in year phenotypes, etc.
Oh, yeah. You mean the marked genes of the different cell type, right?
Yeah, I guess that this would be captured at the cis regulatory elements of the genes that lead to changes in the patterns of expression across genotypes. So I don't know if there are examples of that you're aware of.
I understand. Because in the firstly we analyze this is the genes that you said, like some genes especially expressed in the some cell types and another expressed in the other cell type. Actually, this kind of genes is we care most, is the gene we care most. And in here I show you some case that control the year development is the genes we found, they expressed in the specific locations. So I think, sorry, but I don't understand what's your question. You mean change the expression pattern of these genes?
Yeah, across different genotypes. I guess that what we are all want to see is how this technology could be applied to explore larger numbers of genotypes, and to explore to what extent these patterns vary across genotypes. But it seems like you'll get there at some point. But
You mean explore the specific expression genes in the cell, in some cell type, right?
yeah, no, related to what Eli was saying. If you were to do the same experiment, for example, in other NAM parent lines, would you expect to see a lot of changes in the patterns of expression? Would you expect to see some lines expressing some of your candidate genes at a particular tissue type, and other lines expressing at other tissues, that sort of thing? But yeah, no, I understand. It's a very expensive experiment to do, very complicated to take the tissues, and I guess that the data is not there yet.
Yeah. We didn't take such experiment because it's very difficult to change the experiment of the genes.
Thank you.
Okay, thank you.
Yeah, thanks, Ruben. And just another thing that I realized, Bob Schmitz was here at ISU yesterday and was showing us some data, looking at tissue samples, but then breaking this down to cell types within tissue too. And a lot of genes that were showing up in these cell types, you would miss the differential patterns of, he was talking about open chromatin, but, correlated with expression. If you just did a sample of the entire tissue, like it was very clear that things were popping up just in single cell types. So we'll discover all sorts of new things with this new technology that you're using, Yuebin, that we've missed when we've done entire tissue examples of differential expression.
Yes.
Cool. Well, any final questions for Yuebin? This has been a great Q&A session. But did anyone want to round this out with any final questions? Okay. Well, I think we'll finish up here today then. And thank you so much for a great talk and joining us at a late time at night. Really appreciate that. I know this isn't totally convenient for China. But
It's okay.
yeah, we appreciate you joining us. And happy to have you keep logging in too and join us for this semester.
Okay, at last, I would like to say very thank you for giving us this opportunity to share our work. If you anyone is very interested in our work, you can on this website and you can find any one of us from our lab, and you can contact us for the detail of the experiment or the analysis pipeline. Okay.
Perfect. We'll do that. Great. Well, thanks everybody. Yeah, thanks everyone.
Thank you.
Yeah, and remember there are open spots, and I would love to have somebody sign up for next week. So practice that maize meeting talk. All right.
Okay.
Talk to you guys later.
Thank you. Thank you.
¿Cómo estás? Bien, bien. Solo venía a hablar con